So I was asked to give a follow-on on the ILSI North America workshop because I was there. I reread the cell host and microbe, the references there, and what I'd forgotten from that meeting is we were told to focus on health, and we all focused on disease, and it was tough to get us to really focus on health. And I see some of you nodding because that they're not the same thing, and yet many of us jump right away to the disease conditions. And as Ricky pointed out, there are still many questions related, but more answers than there was at the time. So I'm going to give you an update. And then to follow along what Cindy did, I'm going to give you an international flavor of what's going on the microbiome research, focusing on what we're doing in Canada in collaboration with our European colleagues. So this is a very stark contrast to the NIH with no disclosures. At CIHR, we're encouraged to collaborate with partners, and in my research enterprise, I focus on probiotics and prebiotics in infant formula with a company that's has a probiotic that's been approved for use by regulators in Canada, the first in the world, I think, as far as I know, before Europe and before the United States in using probiotics for humans, and I'll mention that, and a company related to a biotech startup. So the development of the gut develops, as you heard, in utero. It was thought in the past that the fetal intestine was sterile, and there's more and more evidence using genetic techniques that that's not the case. There is bacterial DNA, and it might be culturable. It's clear that the mode of delivery is very important. The microbiome composition and probably function differs widely between babies who are born by cesarean section and vaginally. And the next speaker will talk more about the immune system response related to delivery, so I won't go that way. The diet that the baby receives after birth, whether breastfed or formula fed, and what kind of formula, has an impact on the developing gut microbiota, both structure and function. Where the baby lives in the world and within a setting like the United States in a high or low socioeconomic circumstance has a huge difference in the developing gut and its impact in both the short term and the long term. And what's becoming increasingly recognized is that the microbiome we all have of this generation is probably different than what it was even one or two generations previous to me. And that is because antibiotics have started in the world. And antibiotics we are exposed to them, but so are the animals that we eat. In even very low doses can really impact the microbiome. An interesting work in a mouse model by Marty Blazer, published in Cell in the summer last year, showed that very low doses of antibiotics, subtherapeutic in a mouse, impacted the metabolic function of that gut microbiome in the adult mouse. So huge implications about even low exposures of antibiotics to later life. So some data, just to support what I said in that summary slide. This is work from a colleague, a neonatologist who came from Sweden to spend time in my lab. And it's looking at the diversity of the microbiome on the y-axis over time on the x-axis. And it's comparing diversity in cesarean section and vaginally delivered babies. It's the other way around, the vaginally delivered and then the C-section. And you can see that there's a markedly reduced diversity, at least for the beginning part of the life. And what that impact has on the developing newborn and infant is the subject of lots of questions. We don't have the answer, but there certainly are changes that might impact on the immune system and other aspects of health. This is a study from Denmark showing the same thing, that diversity on the y-axis, chronologic age on the x-axis, that healthy Danish babies' diversity increases over time. The more premature that a baby is born, many premature babies are born by C-section because they're in distress or the mom's in distress, the diversity is much less, and that we think has functional outcomes. But as you heard from Dr. Davis, most of the studies on the microbiome to date have functioned on characterization. It was very important to have that as a baseline. But there is a lot of redundancy, and it's the functional outcomes of that change in diversity that we need to know more about to establish cause and effect relationships. This is an example of antibiotic use, diversity, again in babies, diversity on the y-axis and time on the x-axis. And it shows that there's a loss of diversity in babies who are exposed even to low-dose, single-dose antibiotics. It doesn't have to be a broad-spectrum cocktail, although that might even have more of an impact on the microbiome, not only in the gut, but elsewhere. You heard about the vaginal microbiome, the nasal microbiome, the skin microbiome. So antibiotics can certainly have profound effects well beyond their local. So things that relate to health and promoting health and reducing disease, two sides of an equation. One of them are to look at functional foods. So nature's first functional food is mother's milk. So a large component of mother's milk are human milk oligosaccharides. They function as prebiotics. They promote the growth not of all organisms, but certain organisms that have the enzymes to degrade the complex galacto-oligosaccharides. And so breastfed baby stool, exclusively breastfed, is yellowish, acidic, related to the bugs that colonize that baby, which are mostly bifidobacteria and lactic acid producing bacteria. So we know there is a prebiotic effect and that it changes in babies who are fed formula or upon weaning. And mothers know that right away and parents know that right away. When babies wean, you can see the color of the stool changes, the composition changes, the consistency changes, all related to the microbiome switching to an adult composition. We know that the breast milk has the human milk oligosaccharides, but more and more evidence of beneficial microbes, including bifidobacteria, and bioactive compounds. And I don't want to steal the thunder of the next presenter, but it appears that being breastfed has a huge impact on the developing organism's immune system, both innate and adaptive. So sorry if I've stolen too much thunder already with saying that. So here's an example of work that we published recently in Journal of Nutrition. I was bragging to colleagues at dinner last night at my first publication in a nutrition-related journal. It's a little away from my area, but it's using injury to the gut on the y-axis, so it's macromolecular uptake. And it shows that with different prebiotics taken in a control, there's no impact on the macromolecular uptake. But if you damage that intestinal tract, in this case it's in an animal model, you get a big increase in macromolecular uptake. And prebiotics given to the animal can reduce macromolecular uptake. And it's not related to all prebiotic compounds. It's related to the specific structural composition. So there is and it's dose related and it's time related, and there's a structure-function relationship between the activity of prebiotics and their biologic effects. So this is an example of the interface between health and disease. You heard this in the previous presentation. We know that in many conditions, including inflammatory bowel disease, the common theme is a loss of diversity in the microbiome in the gut. And it's true in other sites, but in the gut in particular. And we think that's related to functional outcomes like reductions in short-chain fatty acids. And one of the, we don't really know what causes IBD. There are studies going on in Canada looking at the siblings, not patients with IBD, but siblings, because they're genetically at risk, and following their microbiome, its structure and function over time, to see if you can tell does the microbiome change occur before or after the inflammation, because that's the key issue, is that they're a cause and effect relationship. That's called the GEM study, funded by the Crohn's and Colitis Canada. The one part I wanted to emphasize on this slide is knowing the issue of antibiotics. There's epidemiologic evidence, so it's associative, it's not cause and effect, but there's strong evidence that antibiotic exposure in the first 2 years of life, studies done in Denmark and Canada, correlate with developing IBD in your teenage years and your adult years. It's early adult years when people get IBD. So again, evidence, it's not direct, but indirect evidence that altering the microbiome early has real profound implications on health and the development of disease. So this is a nice review. I guess you can't see the reference there, but it's in the Proceedings of the Nutritional Society last year, which I like because it's sort of two sides of the coin. On the bottom are factors that are involved in promoting health, like breastfeeding and vaginal delivery, and not being exposed to multiple courses of antibiotics, and having the right genetic makeup, and being in an appropriate environment. And the flip side, the increased risk of disease in the red, is exposure to the converse of what I just described. So we need more evidence that reducing the risk factors in red or promoting the beneficial factors in green actually do have an impact on health and disease. And I'll give you some examples where we think this might have an impact. So this relates to the exercise data. Now, I don't know if extreme exercise is health, so that's a part that somebody else will have to tell me about. I'm not a super triathlon athlete, so I don't look on it as a real health state, but I know triathletes do. So this is a study out of Cork, Ireland, and it's looking at the microbiota composition of the intestine in these extreme athletes versus which are on the right-hand side, and the regular old people and moderate athletes on the left-hand side in the color. And each of those dots is an individual person, and it's a principal component analysis. So the closer the dots are to each other, the more similar they are. And what you can see is that these extreme athletes, that's my word, the article says elite athletes, that's the right word. Elite athletes are different than the rest of us. And so it would be very interesting to study the microbiome of those elite athletes, and maybe you don't have to exercise, just get their microbiome or transfer the bioactive ingredient in their microbiome, and we'll all be Arnold Schwarzeneggers. That's my commentary. So that was on a variant on health. This is looking on a variant of a symptom. I don't know, you would say it's a disease state. It's a functional bowel condition called irritable bowel syndrome. It affects people of all ages, but particularly young, otherwise healthy women. And it's not related to inflammatory bowel disease. This is symptoms of abdominal pain, but no organic etiology described. And the microbiota of the women that have symptoms is in the light gray, in a principal component analysis, and it differs. It segregates completely and is less diverse than the asymptomatic women, same age, same community. And they segregate with a greater diversity, and they're off to the right. The same findings have been shown in children with abdominal pain. It might be a pediatric equivalent of irritable bowel syndrome. And the same thing, less diversity and segregating from normal microbiome in little babies, infants with colic. So again, I don't know that colic is the baby equivalent of adult IBS, but each of these three functional conditions at varying spans along age have been described with a less diverse microbiome. So how can one increase the diversity, and then thinking about increasing that diversity, that has a biologic effect on improving health and reducing either symptoms like abdominal pain and disease I'll come to next. So this is a nice review in Nature Reviews Biotechnology saying you can go from a fecal transplant to using a single strain, a probiotic or a prebiotic as a non-bacterial growth promoter of beneficial microbes, to a bioactive compound that's mediating the effects in the probiotic, for example. The thing that I like about this way of thinking is that there is very good evidence in a specific disease condition called Clostridium difficile infection, and it's mostly in the elderly with a very reduced diversity of the microbiome, that you can cure them of their recurrent infection with a fecal transplant. So that's giving from a spouse or a sibling. But it turns out, and that's what's published in last week's Nature, is that that's not related to the whole microbiome. You can give a specific single organism from the microbiome that recapitulates all the effects of the whole microbiome. So there is a bioactive ingredient. It's an organism in this case, but whether it could be a component from an organism, that's the next step that we need to know about. So this is looking at trials of using a single organism a probiotic organism in infant colic. And this is a meta-analysis plus one trial that's just been published from Toronto Sick Kids, showing that compared to placebo, this single strain reduces symptoms of irritability in a little infant. This is a meta-analysis also from Canada, Paul Moietti, looking at adult women with irritable bowel syndrome. All the studies have been done. The summary odds ratio is the one circled in red. Probiotic strains, multiple ones, but not all of them, and it's dose-related, have an impact on improving symptoms of irritable bowel syndrome in women. That might not look like a dramatic effect, but it's as powerful as any other known treatment for irritable bowel syndrome, and the number needed to treat is only 4. So if you're a health professional, anything that good is worth considering in your therapeutic armamentarium, especially if it's safe, because that is evidence of efficacy. Now, regulators don't rely on meta-analyses, at least Canadian regulators don't. They rely on single studies, and there were two. There's actually 2 products that have been approved, both based on single RCTs. I don't know if FDA would approve or Europe would do the same. But in Canada, they took the, I think, very thought-provoking leadership approach of saying, let's use probiotics in an environment where it's least likely to cause harm. So not little premature babies weighing 600 grams, and not the very elderly at risk of multiple infections. But let's start with adult women who are otherwise well, apart from having abdominal symptoms, this irritable bowel syndrome. And it was based on this is an Irish study, showing there's a dose, the low, placebo's on your right, the white open bar is the low dose, no effect, a medium dose, there's a biological effect, and another probiotic had no effect. So it was strain specificity and a dose response was the basis that Health Canada approved this for use for women with IBS. Now you might be wondering what's the story with that black bar histogram, which is the high dose. Now that's kind of interesting, right? Like, why does, if a little's good, a little doesn't work and medium is good, you think a lot more would be even better. And yet it doesn't work. And the explanation for that is, I think really the power of this study, is it was a formulation issue. The stuff was concrete, it wasn't released, and so rather than hiding that data, the researchers presented it, showed that was the mechanism underlying that absence of effect. So it was actually used by regulators as a positive thing, not a negative. So as a result of these RCTs, in Canada, 2 probiotics are approved. Normally for these kind of events, we only give the name of this bacteria, but because they're not familiar to everyone, we were allowed in this medical education slide deck to give the brand names. These are brand names in Canada, the dose, and how long they're used for. So there are these 2 strains. There have been no side effects reported, although there's not a formal and an informal post-marketing surveillance. So the first half of my presentation, because I'm going to give you a little bit about what CIHR is doing, is the gut microbiota is recognized to play a role in promoting health, that altering the microbiome so it's not as diverse appears to play a role in a variety of diseases, and that if we manipulate the microbiome back towards diversity using prebiotics and probiotics or human milk oligosaccharides, we might promote health and reduce disease, but we do need comparative effectiveness studies. So a little bit about CIHR, which is funding research on the microbiome, along the lines that you heard about by the NIH. But all our research is extramurally funded. We have 13 virtual institutes, nutrition, metabolism, diabetes, and an infection immunity. But the Aging Institute, multiple Cardiovascular Institute, all interested in the microbiome. We ran a Canadian microbiome initiative. It's run around the same time, starting around the same time as the NIH. Funding has gone up dramatically with targeting the importance of the microbiome, characterizing it structure and function, composition and function. And the absolute amount of dollars is much less than you saw for the NIH, but I don't think I really need to apologize for that because we don't pay investigators' salaries off CIHR funding. That's the responsibility of academic institutions and research institutes. So we fund the research, not the salaries of researchers. And you can, whether that's good or bad is a whole nother discussion. So here's some examples of the teams that we funded. There is a group looking at the vaginal microbiome. There's one looking at the developing infant's microbiome and setting of IBD. The one thing I would say about the way we conduct this kind of research is we try not to fund the cohorts that are required. There are Canadian cohorts of children and the elderly. It's called the Canadian Longitudinal Study for Aging. You actually don't have to be that old to enter. You only have to be 45 to get entered in that study. And that was a very, yeah, I know you're laughing that you're now old all of a sudden. But that was actually a very smart thing to do because it means we can do intergenerational evaluation among grandparents, mothers and fathers, and their offspring on their microbiome composition and function over time. And that is the intent of what we'll study. On the international, and we are looking to fund new microbiome projects. I've been talking to some of you about an environments, genes, and chronic disease initiative that we're launching, and we're looking for partners to launch that funding opportunity. We know there's interest in Europe, in the European Union, through these joint programming initiatives on the impact of nutrition on health and disease. It's got this name JPI-HDHL, Healthy Diet, Healthy Living. We've been involved in funding with European countries biomarkers in nutrition, and we're about to launch together with New Zealand, Canada, and Switzerland, and then a bunch of EU countries, a launch on intestinal microbiomics. So the focus of this one, as you heard from the NIH, will be more on not just who's there, but what are they doing? What's the composition? How does it impact on promoting health and preventing specific disease states? So if you're interested in the launch, the funding that Canada is going to do together with Europe, we held this workshop in Vancouver, and I'm happy to share it. I have it on a copy if anybody would like it. So with that, I'd like to conclude, and thank you very much for your attention, and only if there's time, take questions. Thank you very much.
We do have time for a couple questions for the break.
I have a question. So you talked, Phil, about the infant formula, but I didn't, and we know what doses are in there, but I didn't hear you talk about other products that are available in the market and the doses and the type of probiotics and whether they really are effective.
So dosing of probiotics is a critical issue. In experimental conditions and in the human disease states, the doses are orders of magnitude of what you see in fermented foods or in yogurts that you get in the grocery store. They're orders of magnitude higher than that. So there probably is a threshold level that is required, and what the two drugs that were approved, the print was probably smaller than you could see, but the dosages that are used in the IBS products are orders of magnitude higher than in yogurt products that are purported to improve abdominal wellness. I think that's the terminology that's used. So yeah, dose and route of delivery and strain, choice of strain are all critical factors.
I did mention that we had time. We actually have 15 minutes. We can take questions now before the break. Sorry, Brett.
Maybe Cindy, you want to come up?
All right, in that case, I got two questions. My first question is, in the last two talks, we heard about the diversity of the microbiome, yet there are a lot of conclusions that have been presented here about what is affected, what's not affected from cardiovascular and these types of things. So how do you reconcile that given the given the diversity of individuals' microbiome? Number two, speaking as a father of 2 young children who've basically used a lot of antibiotics for ear infections, how do you, you talk about that antibiotics, even small doses, have a large effect on the microbiome, and you had mentioned it may lead to IBS later on in life, et cetera, et cetera, et cetera. But there's a quality of life issue as well for the children. So how do you reconcile those two types of things?
Yeah.
While it may affect the microbiome later in life, I'm not going to let my child go with an ear infection and an ear infection and on and on and on. You know what I mean? So
I certainly do.
if you could comment on those.
Maybe Cindy, you want to comment? So the one thing about diversity is giving a single strain. You don't need to give a fecal transplant to alter the composition of the function of the microbiome. Giving 1 or 2 strains can have a profound effect on the diversity of the microbiome. So that's been looked at. We published that in an experimental model. So when you give an acute enteric infection, you get a spike and a loss of diversity, and the pathogen is there in large amounts. And then as it comes back down, the diversity comes back. And if you add a probiotic or prebiotic, it doesn't have to be multiple strains. You promote the diversity. The issue of antibiotics, life, the good old days wasn't all the good old days. There were a lot of terrible things that happened in the absence of antibiotics, and people's lives are saved using antibiotics. So there's no doubt about that. On the other hand, there's no doubt that antibiotics are overprescribed for humans and overprescribed for animals, and that there are alternate choices to promote, say, growth of animals, right, rather than using subtherapeutic doses in feed, which is done now, for example. So it changes the way that we think about the impact of using antibiotics, because it isn't just about high dose, it is low dose. Cindy?
And I'll continue with what you were saying. Also, there are times when antibiotics are definitely needed, and it does come to a quality of life issue. There are investigators evaluating, should you also, if you have to be taking antibiotics because of specific diseases, should you be taking probiotics at the same time, and would those have beneficial effects then? So it's one of the research questions that's out there.
Yeah.
I wanted to ask about 2 observations that seem somewhat incompatible. On the one hand, transfer of fecal transplant gut microbiota from one organism, particularly mouse, into another mouse, seems to lead the recipient mouse to take on, with respect to growth and adiposity, the phenotypic characteristics of the donor mouse, suggesting that at any point in life, the effects of the gut microbiota on growth are reversible. On the other hand, Marty Blazer's work advocating the idea that antibiotics alter the gut microbiota, which in turn then lead to growth promotion, indicate that when you pulse the mouse with the antibiotic, you largely wipe out the gut microbiota. You then withdraw the antibiotic. The gut microbiota within a month or so recolonizes back to normal, and yet the mouth stays on an altered trajectory of adiposity, suggesting it's not reversible. How do you reconcile those 2 findings?
So, Cindy, I'll start and then you'll go. So one of the things, the talk yesterday about imprinting smell really resonated with me, because age is probably a really critical issue. The interesting thing about Marty Blazer's paper in mice is that it was a very low dose of penicillin, so not a broad spectrum antibiotic, and it did not wipe out the flora. It just altered the composition of flora. And the interesting thing is that had profound effects, at least for a mouse, we don't know about in a human, later in life, and they had increased rates of insulin resistance and adiposity. So I don't think those 2 are at all at odds. I think they just show that timing, like everything in life, is critical.
Yeah,
Cindy?
and I'll also talk about, a little bit, I completely agree with those comments. And also there's a very interesting paper that came out of Ruth Lay's lab a couple of months ago, where they actually took germ-free mice and gave them the microbiota from twins that were discordant for obesity, and then they co-housed them, and they actually found that the microbiota from the lean individual was actually more likely to have a biological effect if they were co-housed it was actually the lean microbiota that became dominant, and that the more ones that got the microbiota from the obese individuals actually started losing weight. So there is evidence in preclinical models to suggest that modulating the microbiota may actually have a role in the treatment of obesity, but the science isn't there yet. But it's where it potentially will be going in the future.
Yeah, totally agree. Joanne?
Thanks for a wonderful talk. I was wondering about the antibiotics, I think I understand, but what about other chronic medications taken by very large proportions of the population? And what about things like dietary supplements? Is there any evidence that they have ill effects? I suppose Metamucil does, but what about
Yeah,
other things?
so Cindy, can you help me? I think it's an excellent question, but I don't know the answer. Your Office of Dietary Supplements, right? So,
Johanna's Office of Dietary Supplements also.
Well done.
yeah, I don't know. Do you? It's
And
got to be looked
I
at.
actually really don't know either. Sorry.
Yeah, good question. Very good question.
My question is maybe to both of you again. The slides that Cindy showed with the difference in the microbiota, whether the diets were mainly carbohydrates or not, or not. I was wondering if you have any insights or if you know of any ongoing research, because there is a huge trend against carbohydrates in this country, and people are dieting with selective diets with no carbohydrates, high protein, you name it. I mean, all the combinations are possible in the dieting world. Are there any research that are looking at this effect of dieting and how many days to recover a healthy microbiota if the individuals revert to a balanced diet?
So the answer is protein and fat as well as carbohydrate have been looked at, and they alter the composition of the microbiome and sometimes the function. So I showed one, I forgot to say it, but those elite athletes, the diet difference between them and the regular is that they're on a lot of protein. They take really high protein diets. So it isn't just the exercise, there's a confounding variable in there of a lot of protein. Gary Wu, who is funded through NIH, has done a lot of work on high-fat diets and effect of the composition of the microbiome. And Jean Chang in Chicago has looked at the high-fat diets, altering bile acids, promoting the likelihood of inflammation in an animal model of IBD. So I think all components of the diet in those big broad categories, not the specifics that were asked about in the previous question. Joanne, Cindy, do you want to add comments on?
No, I think you answered it well.